You're listening to the Animal Heartbeat. My name is Kieran Bourget.
Hi, and I'm Jose Logumatos. This is the world's only podcast dedicated to veterinary cardiology.
Welcome, everybody, to this brand new episode of the Animal Heartbeat. And as part of our Cardiology Icons series, we are honoured to welcome Professor Kate Meurs, who is an ACVIM boarded veterinary cardiologist turned geneticist. And she's also Randall B. Terry Jr. Dean of NC State University. Jose and I are honoured to have you here, Kate. Welcome to Animal Heartbeat.
Thank you. I'm so honored to be here, and I'm certainly not an icon, but I'm delighted to have this opportunity to talk to both of you and anyone else who's listening.
Fantastic.
Thanks very much, Kate, really. We thought we would start with the basics. Could you share with us when and how did your interest in genetics begin?
Yeah, it's great. I was thinking about that. Really, it started here when I was a small animal intern on cardiology. So I thought I would want to become a cardiologist. And in those days, so early 1990s, we saw a lot of Doberman pinschers, and we had no pimobendan. And so I really remember to this day loading this beautiful Doberman into a station wagon with this woman and her small children and telling her that this dog was probably going to live another 4 to 8 weeks. And here was some digoxin, and it might make him nauseous, and he might vomit, and good luck. And I thought, this is awful. If I have an opportunity, this is something I would want to work on. And when I did have that opportunity as a resident and a graduate student, I really thought about what is it about cardiomyopathy and Dobermans that I could have a role in and that is interesting to me. So, you could develop better drugs, or you could people were starting to do surgeries and things, but those really weren't so interesting to me. And when I was reading about causes of dilated cardiomyopathy in people, they were just starting to have breakthroughs in genetics. And I thought, clearly it's in Dobermans. I mean, other breeds get it, but there's something unique about this. And so I thought naively, if I could just find the cause to the genetic cause, then maybe we could remove it from the breed. You could breed it out. And that's where I was very naive. But I do think understanding the cause does help you do a better job managing and treating those. So that was really what turned me on to genetics. And I think when I thought about all those things as a really young clinician, where do I want to make a difference? The thing that grabbed me was genetics, this concept that in people they were discovering that a single base pair change could change your life. If you were born with that, that was the impact of that. And just for me, I could almost salivate about that. Like that was like, wow, that is so fascinating. And I could look at pedigrees and DNA and just stay up all night looking at it. So that's how I stumbled onto it. But it turned out that I like puzzles, and genetics is a puzzle. And I loved that.
It's such an attractive proposition, isn't it? Being able to say, oh, there's this thing that causes it, so let's turn that off.
Yeah.
But it's just not transpiring to be how things work, is it?
Yeah, I was young and naive.
But I love the idea. And I think the more information we have on exactly what's happening on a protein level, on a molecular level, the more weapons we can develop to try and deal with that disease. So tell us a bit about the journey there. How might we try to identify a genetic abnormality that's associated with a heart disease? And then how do we go on to prove that abnormality is important or not?
Yeah, it's so important to make that link between the gene and the biology, I think. And so, we always start, if somebody comes to us, and we've had the pleasure of working on a few of these things, somebody comes and says, I think that this might be familial. I'm seeing it I've got a family, or we're seeing it more in a particular breed. We generally start with trying to collect a number of really good quality DNA samples. And in this day and age, we generally do whole genome or exome sequencing and just essentially sequence that whole dog or cat, compare that sequence to a really large group of what we believe to be unaffected animals of many different breeds. So the current canine pipeline that we use, I think, has over 3,000 or 4,000 different dogs of all different breeds. We know, and all of those have been genetically sequenced for various diseases. So I'll go through and I'll say, I don't want to use any of the Great Danes because who knows, maybe they have cardiomyopathy. I'm looking at a cardiomyopathy. So I kind of narrow down what the controls will be, compare them, and then identify variants that are just in my affected animals. But then as you implied, it's almost you're building a case, just like with diagnosis or when you're tracking down a criminal. Yes, you found a variant, but do you believe it is high impact? Does it change the amino acid that's produced? Is that amino acid highly conserved across species at that place in the protein? Do you believe that this variant could muck up the protein? And then can you make a story that if you muck up that protein, you could get this disease, whether it's arrhythmias or heart muscle disease or development of congenital valve disease, whatever it is. In an ideal world, you have nice tissue samples that then you can also do the biology and say, here I've proven that this variant changed the protein, and here in the affected animals, you can see that the protein is wacky. But it's hard to get all of those things because sometimes you don't have tissue samples. So then you have to really make either a very strong statistical story that the odds are that it's so rarely in any other population or in what are believed to be unaffected animals of your breed, or you really make a strong story for its biology. But we all see examples, including my own, where sometimes you get really excited and you think that this is it. But then as you start looking at it in a larger population, it doesn't continue to play out. So, and there's a lot about genetics I think we don't even know. Certainly individuals can carry causative mutations and not get the disease. That doesn't mean that the mutation isn't causative. It just may mean that they have other protectors or something. So it's hard to make a black and white story in some cases.
Does it help to compare the sequence of the affected animal to related animals to try and remove some of that background noise, or is that more complicated? Forgive my ignorance if that's a stupid question.
No, it really, I think it depends a little bit on, it's a really good question. I think it depends sometimes on the gene pool, how big that breed is, how obvious the disease is. Let's take mitral valve disease in, I don't know, let's just even say in Cavaliers.
Something super easy like that.
Yeah. So, there's a variant that even as you look across all the other Cavaliers, even dogs that you think don't have this disease, maybe they actually have the variant, or maybe they are lucky enough they didn't show the disease till 13, and you evaluated them all at 10 or 11, thinking if they don't have it, if they're a Cavalier and they don't have it by then, they're clear. So I think those things are like French bulldogs would be another example. They have a very small gene pool, and so everybody's pretty related. And so comparing their own family members can be challenging. I think we did better when we looked at Rhodesian Ridgebacks. That was a family that Steve Rosenthal and his colleagues brought to us. They were dying of sudden death. And those family members, if they had arrhythmias, that that was really helpful. The unaffected, if they didn't have an arrhythmia, their DNA was a good control. If they had an arrhythmia, it was very clear who had the disease and who didn't, and you could look at that across their family.
I think also something interesting for our listeners to understand is how the genetic pipelines and how your approach has changed over time. Because of course, even when I started my PhD, 10 years ago, 8 years ago, whole genome sequencing was very expensive. So we would do target gene sequencing. We rarely could afford whole genome sequencing. So nowadays, whole genome sequencing is very affordable or even cheap. So easy to get. And you were also talking before that we were looking at the exomes and the coding regions of the genome. But nowadays, and as you've shown, for example, with your striatin mutation, with your PDK4 mutation, you may be looking at non-coding regions of the DNA, right? The intronic areas, entrance UTRs. So how has that changed? Because is it nowadays that you just do whole genome sequencing and then the tools just help you filter what are high impact, moderate impact, low impact, and that's it? Doesn't matter if it's in the coding or non-coding regions, or you still need to carefully look at coding versus non-coding regions?
Yeah, it has gotten so much better. When I started, we didn't even have a published feline genome. There was a little bit with dogs. Everything was so rickety in the beginning. But so these days, when you run it through the analysis, it will say these are the non-coding and these are the coding regions. And the low-hanging fruit is the coding region. So you start there. Is it exonic? Is it splice site? Is there a big deletion? But there's so much in non-coding areas, in big intronic regions, that even in human genetics, we still don't understand. So I think we have many examples where we really believe something is likely to be inherited and there's a genetic mutation there, but we cannot find it because it does not appear to be in the coding regions. You can find a few things that look of interest in the non-coding regions, but it's very hard to make a biological defense of that argument that is causative, because we've come a long way. The computer analysis now can say, this is coding, this is high impact, these are non-coding. But there's so much about what happens in the non-coding part of the genome that people really don't know very well. Is it histones? Is it methylation? Is it, the way it's chromatin or folding, all those things that are very frustrating, because you think we know it's inherited. We've spent a lot of time and money looking and just haven't found anything that we can make a story for. And one of the things I've found kind of frustrating about genetics is that I think it's easy to be cynical about it and say, especially if you can't make a good story for it, it's understandable to say, well, that can't be the causative thing. We should ignore that. You don't want to overcall it and have pet owners out there rapidly screening and removing something that's not important.
Which is a problem, right? That happens,
It's a big problem. And so finding that fine line, and I may riff on that topic for a second, is I think it's really problematic. I think breed associations, and I love them all and they're wonderful. Either they tend to go one of two ways, very black and white. There's a causative mutation. We should stop breeding any animal that has that, which I do not agree with. Or the other way they'll go is, we don't believe this because not all animals that have it ever get sick. So that's a worthless mutation and we're not going to use it at all. So a lot of genetics, I think for a lot of clinical genetics for veterinarians, I think needs to be around education and educating our students, it's not black and white. It's a useful tool, but used incorrectly, either one of those ways can really hurt the pet population.
And I think, we're sitting here as very privileged people. We're well educated, we're intelligent, and still we say, oh, it's a little complex.
Yeah.
And if we're saying that,
Yeah.
then there are many, many pet owners out there, breeders, carers who, of course, they struggle to understand it because we do. But I think we're just comfortable with the uncertainty.
Yes.
And I think many people aren't.
And it's also become really accessible, right? I mean, they can pay $60 to send in a buccal swab and run all sorts of tests. And some of the testing companies, I'm not finding fault one way or another, but some of them, their business plan is the more tests you run, the less expensive it is. So maybe they started out there thinking they're going to run for a cardiomyopathy mutation, but now if they do 5 more tests, the price comes down a little. So what else should they run? Oh, maybe blindness. What if, even if their breed never has a problem with that? So then they get this information, which is very confusing and may not even be relevant to them. And then when maybe a mutation comes or something comes back that surprises them from that, then that's when they say, well, this is worthless. I spent this money, let's not even bother, which removes, I believe, a powerful tool, a way we could be helping decrease animal suffering in a proactive way.
Kate, one of the ways, if we carry on understanding how we study cardiomyopathies and how we then discover a mutation and then look at how that mutation affects the phenotype and the correlations between genotype and phenotype, we'll start talking about animal models. Could you give us your thoughts about what are the strengths and limitations of using mouse models versus large animal models such as cats and dogs?
Yeah. I heard Jose answer a very similar question the other day, and I could not answer it any better, but I will attempt. I mean, I think, I think mouse models are fine. There's nothing wrong with mouse models. They're a good starting point. I think particularly maybe for drug studies, a good place to start. I think that when it comes to spontaneous heart disease, looking at animals that have a naturally occurring model, a naturally occurring example of that is a much closer model than a transgenic one. So if I'm a human researcher studying familial cardiomyopathy, I do think there is a lot that can be learned from looking at natural animal models of that disease. And I think the benefits of that does relate to the genetics. So, if you're studying familial cardiomyopathy in humans, there's a lot of genetic variation just in the 3 of us that make us all so different. And so that makes looking at what is the impact of diet or exercise or even other genes across humans hard, because we have so much variation anyway. However, if you use a purebred, naturally occurring animal model, like let's say Doberman Pinschers, the background genetics of Doberman Pinschers is similar, more similar than 3 humans, because they've been line bred by people that are breeding for a certain look and a certain behavior. And so you are removing some of the genetic background noise. And I believe, therefore, natural animal models of familial disease are really good models to study what are some of the other environmental impacts on familial disease, and also even other, an easier way to tease out what other genetic factors might make the disease more severe. So for instance, let's talk about boxers, because we haven't talked about them very much. If you wanted to know why some boxers get really ugly arrhythmias and some just have abnormal Holters but never get sick, and they both have the same genetic mutation, because they're overall genetic background is similar. They're all boxers. Maybe they're American boxers versus English boxers. Maybe there's a little difference there, but that has removed some of the noise. And so it's a little bit easier to look for, are there other genetic factors in these two pools? They all have the mutation. They're all boxers. Can I find other things that might separate the dog with severe disease or the dog with mild disease? That's really hard if you look at 1,000 human beings with different, with cardiomyopathy that come from different backgrounds.
Because now that we talk about large animal models, when you describe the first disease-causing mutation in cats causing hypertrophic cardiomyopathy, so the A31P variant in a myosin-binding protein C, that was, to me, well, a really huge breakthrough and a huge milestone, because then fast forward, that has allowed Dr. Kittelsen and now Dr. Stern to build this cat research colony, which is absolutely unique, right? And without your work on identifying this mutation, this would not have been possible. And this colony is only getting more and more interesting, right? Because now the colony has just been used to study cardiac myosin inhibitors, which might be the biggest breakthrough in human medicine and potentially feline medicine in the treatment of HCM. So can you tell us a bit the story behind this mutation? How did everything start, and how do you end up identifying this causative mutation in cats?
Yeah, so first, you're so kind. It means so much to me that you say that. I don't feel that, but I am grateful that you would say that. You know, so a little bit of history. I left here as an intern thinking I thought I might want to work on genetics a little bit. I went to Texas A&M and worked with Matt Miller, and you could do that program. You could do a residency PhD, and Matt's an amazing guy who, if nothing else, just always encouraged me to be happier than he was, which is very hard. So it set the goal very high, and I love him for that. And so, but he also gave me a huge amount of freedom to study whatever I wanted. So I was going to work on Dobermans and genetics. And in those days, there was no feline or canine genetic researcher at Texas A&M. And so Matt and his wife, Terry, arranged for me to meet with this guy, Jim Womack, who was a world-renowned cattle geneticist. And he said, sure, Kate can use my lab. I'll show her basic techniques. Everybody in his lab was working on cows. He was world-renowned, lovely man. And he was working on genes that improve milk quality in cows. Anyway, so I started working on Dobermans, but I had to collect the Dobermans. I didn't have any samples, so I was screening the Dobermans on Saturdays and collecting blood samples. And Matt arranged for me to go to Baylor College of Medicine, which was 2 hours away, and find a human geneticist there, a guy named Jeff Tobin, who was a human cardiologist that worked on dystrophin. Anyway, I just randomly knocked on people's doors, a cardiologist that studied genetics at Baylor, and this guy, he had a picture of a duck on his door and that his daughter had drawn, and he opened the door and I said, I'm a veterinary cardiologist in training. I wasn't even from Texas A&M, that vet school down the road.
And I'm really
ducks. I'm really interested in genetics. And he said, sure, be happy to help you, blah, blah, blah. So I would go spend summers working with Jeff. But again, I didn't have any samples. So Mark Kittelsen had been very hungry to make this breakthrough. And he had a breeder that had given him a few Maine Coon cats. Goldie, I think one of them was named. Mark will listen to this podcast and he will correct me, I'm sure. And so Mark, she'd given him a couple of Maine Coons and he wanted to breed them to prove that it was inherited. And I believe he took them home. They wouldn't breed in the cat colonies at Davis because they had been show cats from this lady's house. And so and this would not be legal, and probably it wasn't in those days, and I'm going to get Mark in trouble with Davis, but UC Davis. But so he took them home to the Kittleson household, and there they were much happier and started breeding and had
You
kittens.
put on some romantic music, dim the lights.
His wife was okay having these cats around. So Mark had a graduate student who was going to work on this. This is my memory of it anyway. And something happened. The student quit. Maybe they got sick waiting for cats to get, produce kittens or whatever they needed to do. And so right around that time, Mark found out, or I bumped into him somewhere at ACVIM perhaps, that I really had this interest, and I'd made some connections at Baylor, and he said, I'll send you my samples. So it was all sort of luck and timing. His person had quit, and all he had these cats, and he had DNA samples, and every time one would die, he would pluck its heart and put it in the freezer. And Mark was amazing to me. Every one of those people, that's how, as you guys know, these things work. Matt Miller, Jeff Tobin, Mark, all of them opened doors, said, sure, you want to work on it. So he gave me the samples. There was no feline genome, so it was really candidate genes. I would create DNA primers based on the known genes in people. And we started with myosin heavy chain because that was the most common cause. And it's really big, not that big compared to other things, but 40 exons, I think. So we'd gone through all that, nothing. And then just myosin binding protein was next. But also, Mark had some heart tissue that I did some Western blots and things, and myosin binding protein C was abnormal. I had originally thought it was reduced subsequent. I was sure it would be a myosin heavy chain mutation, and myosin binding protein C was reduced just secondarily to it, but it was the other way. So it was just all of those people opening doors.
I think veterinary cardiology is a community where people are quite good at lifting one another.
Absolutely.
And opening those doors. So it's interesting to hear that. We're actually hoping to have Dr. Kittleson on the podcast at some point. So I think he may have a different version of this story. I feel a sort of more approved by the Kittleson household tale.
He will certainly have more about the woman. I know there is more history about the person that gave him the cats. I think that took him a while to get those cats out of her arms, but he really wanted to do this. And all those people really were so helpful. Mark has been amazing.
And I agree with Jose about the impact of this. I mean, it's huge. And I think whenever we find something like that is a big sea change, we found a mutation. This is going to be the first of hundreds. But it's not really been like that. So do you have any thoughts on why it's been so challenging in cats to find the 1,400 mutations that we have in people to do with this disease phenotype?
I think I think it will be like people, that there are many. So I would say ragdolls, we also were very lucky with, because ended up being the same gene. We started there because ragdolls look like Maine Coons, so we thought it would be the same. Oh, they're big fluffy cats, got to be. And I think the early thought was it would be one mutation. But even the ragdolls taught us a lot, because same gene, but those mutations are in incredibly different areas. And I think the ragdoll, I felt very good about that example as well. And here, you've done a lot of work with those ragdolls too. And I think what that really taught us is, here's a cat. You would think cats they're just cats, but spontaneously develop two different mutations in the same gene. So it's not the founder effect. And we used to think, there's probably one very important cat out there that broke into all these other cats. And so, no, if you look at ragdolls and Maine Coons, clearly there's some reason that they develop two different mutations. Is there something about that gene that is more mutable or something. And so I think when we look at the overall cat population, that it's probably not so different from people, and that there are very few, if you look at human beings, there aren't all that many founders that went on for many, many generations. There's a few examples from Holland, which is what I have family from. So, but most of them are small families that spontaneously developed their family's mutation in its couple of generations. So I imagine that cats will be the same, and people are probably identifying them as we speak, but it's difficult to make the biologic story because we prove it either by sheer numbers. I have lots of this breed and they all have HCM and they all have this mutation, and we don't have that many examples of feline breeds where there are a ton that are affected. Or we make the argument, we make the case that we've demonstrated the biologic, here's the tissue. And often when, I mean, Joshua and his colleagues at Davis just finished a study looking at a lot of them, when they're all many, many, many different mutations, you would have to have many, many different chunks of heart that you can show, okay, I had a mutation and I had its heart, and now here the protein is abnormal. So to make a long story shorter, I think that similar to humans, there are probably many, many causes, but we don't have the large numbers, and we often don't have quite the large enough budget to have lots and lots of hearts and all of this analysis. I think that's my best. So I think from his stuff and other people, unless you've got a specific breed, British short hairs or something where you're really seeing a lot of it and you have tissue and really good quality DNA, that it will be hard to make those stories. I think a more interesting, not that that's not interesting, but another interesting question might be for both people and cats. Why do they develop so many different mutations, all which lead to the same phenotype? So is there something highly mutable about sarcomeric genes? And if you have a mutation in a sarcomeric gene in your skeletal muscle, you're okay. But if it's in your heart, you develop HCM. Or they make mutations in those genes at the same rate as everything else, but there's something funky about cardiac genetic repair. Because generally, your repair mechanisms when you create a mutation most of the time should fix that.
Something else that you already touched on it, and what really excites me, I think it would be cool if we have a monogenic disease, autosomic dominant, and then we do a genetic test, we find a mutation, and that's it.
Because that's what we learn in school, right?
Yeah, exactly. Right in school.
We learn that and we go, oh yeah, this genes and the mutation does this thing, and
exactly.
That can't be so hard.
It's all fake. And what I think is really, really cool are these cats, humans that share the mutation, and then we don't know what will happen with the phenotype. The phenotype is very variable. There's a huge phenotypic heterogeneity, and some cats and people will never develop the disease. What I would love to hear from you is what you think is affecting the penetrance of, for example, ACE31P or the R820W in the ragdolls. Do you have any ideas of, we talk about environment and so on, but we have not yet identified a clear environmental factor that might affect the phenotype, or an epigenetic factor. Maybe that would be more difficult. But what are your thoughts on that? What might be influencing the penetrance of these mutations?
I love this because when we figure this out, and somebody will eventually, that will be in an ideal world, somebody comes in with a new pet and it has the mutation, and you can say, do these things, don't feed it this or whatever. So I think this is the most important question. What is those triggers? I think in Maine Coons and Ragdolls, certainly at least being dominant, homozygous increases your penetrance. Maybe a little less sure if that in Dobermans, but also the case in boxers. So some examples where being homozygous for it impacts penetrance. But beyond that, I think the best example, is ARVC, at least in people and in mouse models, they've shown examples that extensive exercise increases penetrance. So if you take a mouse and you give them a desmosomal mutation and you make them a triathlete, then those mice develop disease sooner. And so for me, I don't know if this is right or wrong, but when I saw cases, would tell boxer owners with these mutations, particularly if they were homozygous, that I didn't think this dog should be their running buddy. I mean, boxers love to run and play, and if they want to chase the squirrel, that's fine. But I felt that maybe it wouldn't be good for them to go jogging 3 miles every day, because if you've got a faulty desmosome that, constantly flexing that desmosome, maybe it does wear it out. I think this is a really interesting question, and I don't know the answer to it. I think it's really important. We should be able to start working on some of these things, particularly in pure breeds, because we have less the other genetic noise. But boxer is the one that I have the strongest feeling about, aside from the homozygous thing. I mean, it could be even diet. There was something else I wondered. I wondered a little bit about PDK4, and we played around a little bit with metformin, that if with PDK4 that maybe they weren't getting enough carnitine or enough energy into the mitochondria, and that maybe if you look at the biological source of the disease, you could find some reasonable mechanism to tweak the biology and alter the phenotype.
Because that's the other way of using the knowledge of the genetic variation in clinical practice, isn't it? It's not necessarily switching the gene off or correcting it somehow, but adapting how we look after that animal or tailor treatment in a sort of individualized medicine.
Absolutely. I think that is, to me, moving from my naive days. I've given up on turn it off or breed it out, because it's often so much in the breed, or it's a
Very
small
challenging
pool and you can't do that. But if you could say, even today, without knowing the penetrance, if we can say, okay, your ragdoll cat or your dog, you wanted to have a purebred dog or cat, you spent a lot of money, and it has this mutation. We know your animal is at risk, not a huge risk, maybe a 50% risk. So let's meet the local cardiologist. Let's follow this one closer than we would. And so for $30 US, you can run a genetic test and know whether this purebred animal at risk of cardiomyopathy has that mutation. And now, at the very least, we can monitor it closer, develop a relationship with the cardiologist, perhaps start it on pimabendin if it's a dog, as soon as the Doberman starts to have early dilation, because we're watching it sooner, we can treat it. So I think that, to me, is the real power of genetics, is being able to move our practices to be proactive from being reactive, where we have the sick patient in front of us.
Yeah, I think, we would all certainly look at an echo in a dog we knew to be high risk because of a genetic substrate. We look at it differently, wouldn't we? We'd take those equivocal measurements much more seriously, rather than saying, oh, I'll come back in 6 months, we'll review. Yeah. We might say, okay, we're going to call your dog affected, actually. the challenge with that individualized medicine approach based on genotype is that it seems to be, from the data that's out there, these polymorphisms have different effects in different populations. So what I mean really is whether PDK4 is causative in a European population of Dobermans versus the US population of Dobermans. And to mention the boxer, the same with the striatum mutation. Is it causative or is it co-inherited? I think that's a fascinating area because these dogs are similar, but are they similar enough across the world that we can apply the findings across these different populations? What are your thoughts? I'd love to know.
So I think that PDK4 generally causes mild disease. It's the first one we found. I do think it's relevant. I believe in PDK4 because it truly exists. We found that the muscle was abnormal. It fit the biological story. I think the type mutation in Doberman tends to be much more strongly associated with significant disease.
Interesting.
So I feel pretty strongly about that. I feel similarly strongly about striatin. I don't know. I've never compared the UK dogs, and I don't really know if there's anything different about their phenotype. One could argue that's interesting about it being co-inherited. I think it's hard to make that argument looking at us and say it doesn't have an impact looking at the biology. Clearly is a desmosomal protein. The immunofluorescence of those dogs is definitely abnormal. So I think if it was just inherited, the association of the immunofluorescence findings to the other desmosomal proteins, I would expect to be normal. So, but that's not exactly, I think your question was more about how does somebody explain why a mutation causes or leads to the development of disease in one population, but is not in the other. PDK4 certainly has been found in other breeds of dogs like Chinese crested, and they never get, to our knowledge, don't get a lot of dilated cardiomyopathy. So I would say that my guess in those cases would be that it is most impactful in a certain genetic background. So if you have a type mutation and you're in a mixed breed dog, that dog is much less likely to get dilated cardiomyopathy. And I would argue, I think we rarely see that in mixed breed dogs. If you run a genetic panel, they don't, it's unlikely that they have Titin. So I think it takes the combination of having the mutation and that similar background in which the mutations are found. Now, whether that's because that breed also has other enhancers or I don't know what it is.
You mentioned earlier the idea of protective genes or some sort of modification.
Yep.
Yeah, that's really interesting, isn't it? Because maybe the European population of dogs has some sort of protection on top. Certainly the phenotype in the boxer dog that we see, they're more DCM than arrhythmias. They get some bad arrhythmias, but they're nothing like the US population based on colleagues I've spoken to in the US.
Yeah.
They say that they see these boxer dogs with horrendous arrhythmias, nightmarish electrophysiology.
Yes.
And we just don't see it in the same way.
Yeah, very interesting.
Something else that I think is really cool are like double hits, compound mutations. So those genes, of course, if you, instead of having one variant, now we have two variants that are causative, these may awaken the phenotype sooner or make the phenotype nastier. As you described PDK4 and Titin in Dobies, do you have any data or have you heard of any reports of when dogs are affected by both mutations, if they have a different phenotype, if the phenotype develops earlier or is more aggressive?
Yeah, Joshua Stern and I looked at he had some at Davis, and We did. So what we found was that it wasn't uncommon in true DCM dogs to have both. I think 50% of our DCM dogs that we had between us had both mutations. I don't think we were able to statistically say that they'd gotten it younger or we couldn't really pull out anything along that. But I think also what make those type of studies hard, it was a retrospective study and it was small, and some dogs had started on pimabendin earlier. And so I think the compound mutation is a very interesting aspect. And I agree that I think that you would think that it may cause worse disease, but I don't have evidence for that yet.
So some of your most recent work now, Kate, has been looking at dogs with sudden cardiac death, which is also a very exciting field and has not been really well explored or studied in veterinary cardiology. So you recently published mutations affecting Rhodesian Ridgebacks and English Springer Spaniels. In the English Springer Spaniels, you describe a mutation associated with long QT syndrome, of course, a big cause of sudden cardiac death in humans. Can you tell us more about it? How that happened, those studies? And what I also think is really interesting is, do you think sudden cardiac death in dogs and cats is underreported? I think there is mixed feelings about HCM in cats and sudden cardiac death. I think some cardiologists will think that's not a thing at all, that doesn't exist. Yep. So, and others will feel, no, there are certainly cases of sudden death, but we are not aware of them.
Yeah, I love this question. I do think that sudden cardiac death is a specific phenotype, and I do think it is underreported. I think it's underreported because there can be other causes of sudden death. And so unless people identify some other link, oh, I've seen a bunch of this breed of cat die of sudden death, then I don't believe we put it together. And that's what happened actually in both of these examples. So, well, I'll talk first about the Ridgebacks, is that was a situation in the Virginia area where I think a breeder had noticed that she'd had a few Ridgebacks from a family that had died suddenly. And she had started taking in the parents into Steve Rosenthal, Jess Wiedemann, and some of their colleagues at CVCA. And they echoed them and picked up these ventricular premature complexes. But none of the dogs in those families were ever symptomatic. They just dropped dead. And on necropsy of the initial ones, necropsy is normal because they only have an electron microscopy abnormality. So I think in the original cases, the breeder had thought, maybe the dog bit an electrical cord and it was electrocuted, or did the neighbor poison it? There were these type of things. And so it wasn't
GDV in the breed, or, all these things.
Yes. And so it wasn't until somebody noticed an association, because they were the same breed and they were also the same age. Those dogs were all between, I think, like 10 months and 24 months. So that, as they shared stories, my dog died. Oh, that's interesting. So did mine. Then they made that association. And the same thing happened with the English Springer Spaniels. And those also, it was sort of a breeder that had noticed that and thought that there was maybe something here, and took them in to see a cardiologist, Sophie Jesty, who reached out to us. So, but I think so often in sudden death, the animal died, owner calls their family veterinarian who says, oh, that's awful, I'm so sorry, and they bury it. And unless they share that information back to the breeder, and if it's a one-time thing, and there could be other things, heat, cold, electric, ate a child's toy, whatever the story that people make, particularly if the family veterinarian does a quick necropsy and doesn't find anything, maybe they send off a few tissues that come back normal.
I think it's very challenging in those cases to get a good family history, because either there's not there's no contact with siblings or parents, or maybe the breeder just actually doesn't want to engage with it. If they do, fantastic. There's a whole resource there, but I think that's very hard in my experience anyway, of trying to chase those cases.
Yeah, we have just experienced that for the very first time in my research. So we had I think sometimes people just don't connect the dots, as you were saying. There are these clouds, these cases that happen. People think that's completely random, and then need someone to raise that question for people to start connecting the dots. We just had a breeder saying that she has been seeing young Labradors dying suddenly. And then as soon as we put out a call asking if anyone else has seen young Labradors dying suddenly, we have been overwhelmed with cases across the world, from New Zealand, the US, across the UK, of owners, carers that will say, oh yeah, indeed, my 1-year-old dog died during sleep, and 5 years ago, 7 years ago, but no one made a big story of it or didn't know what happened. So it's really, really interesting. Really interesting. And
Jose, that I've seen that one with arrhythmias, horrible arrhythmias, that I thought this dog has to have a channelopathy. And the dog died suddenly before we could get any sampling, or anything else.
Yeah, really interesting. Really interesting. This has been fantastic. We could keep speaking for a long, long time, but we are mindful of your time, Kate. So thank you very much for joining us today.
Yeah, thanks, Kate. It's been amazing.
This has been fabulous. So first, this has really been an honor. Thank you so very, very much. I really, really so appreciated this and loved this. I could talk about genetics endlessly,
Well, so could we. Bombarded with questions.
The final message, and this is what I try to talk to veterinarians and our students about, is I do think there is power to genetics, that it's a really important tool that we can use as veterinarians to be proactive. And I do think the education aspect is so important, even to our clients, because if they feel that it's not a valuable test and they've wasted their money and it didn't predict, they thought it was going to predict exactly a certain way and it didn't, they will stop using it. And then we lose this powerful tool. And I believe the power to it is early prediction, which allows us to follow and intervene sooner. And now we know at least of a few examples like pimabendin, where early intervention can make a difference. And the more we study that, the better we'll get about exercise, diet, who knows, fatty acids, other things. And so I think genetics can be an incredibly powerful tool to be proactive and decrease animal and human suffering, people who suffer from the loss of their pets. But we have to use it the right way. And I think we have to talk to pet owners and pet breeders who are using this tool. That's not a black and white test. It's one more bit of information to help you and your family vet and your cardiologist make decisions so that they use it correctly and don't become disappointed and disenchanted and stop using it. And that's my little soapbox on power of genetics. So thank you, guys. It's been a great pleasure.
It's a great soapbox to finish on. Thank you. Fantastic to talk to you. And thanks, Kate, for joining us on the Animal Heartbeat.
Talk to you again sometime soon. Thank you for doing this. It's really a wonderful thing.
Thank you very much, Kate.
Thank you. Thanks so much.
Bye-bye.
You have been listening to the Animal Heartbeat. Thanks very much for our listeners.
Keep in touch by finding us on social media or sending us an email on animalheartbeat@gmail.com. Please remember to like, follow, and share to help us find new listeners. And make sure to check out our previous episodes wherever you get your podcasts from or on our YouTube channel.